So NRG1 gene fusions are oncogenic driver events. They actually occur across different malignancy types. The overall incidence is low. It's well under 1%, but they are important to find because now there are new therapeutic options for these subsets of cancers. And the approval of drugs here really is tumor agnostic. One of the challenges, though, is that in addition to being rare, even when they're present, these fusions are difficult to find. The fusion itself comprises the NRG1 gene, which is a massive gene, largely intronic. So if you're doing DNA-based sequencing, you're probably lacking the coverage depth to find these fusions. And RNA sequencing gives it a clear advantage in terms of detection. In lung cancer, again, less than 1%. We see a lot in lung cancer just because lung cancer is so common and because we do sequencing in lung cancer very frequently as well. Ben, in pancreatic ductal adenocarcinoma, really it depends on the KRAS mutation subtype, correct?
Yeah, and for sequencing in pancreatic ductal adenocarcinoma, we have a variety of hurdles. One is there's often not a lot of tissue for profiling. And two, there's historically been a lot of resistance to profiling because we had no actionable targetable alterations. We essentially had five chemotherapies, none of which were molecularly targeted. The one sort of newer drug that was the exception to that was olaparib, which is approved for folks with germline BRCA1/2 mutations following disease control or response to platinum-based chemotherapy. So many folks were saying, why should we bother sequencing these tumors if there's really nothing we can offer patients? But now it's sort of the opposite is true. So there's this about 8 to 10% of pancreatic ductal adenocarcinoma samples that are KRAS wild type, and we think that population is heavily enriched for specific targetable alterations, things like BRAF, HER2, but also NRG1 fusions, where it's seen at about probably 1.6% as opposed to less than 1% in the vast majority of other tumor types. We also in pancreatic cancer now are very excited that we're part of this kind of KRAS revolution of new targeted KRAS inhibitors. So we now hear not only if there's a KRAS mutation or not, but we actually care now which one, which is now mostly still in clinical trials, but the hope is that we may be having a whole host of new targeted therapies, not just for NRG1, but for other more common alterations like KRAS mutations. In pancreatic cancer, we've had a dearth of drug development, largely because we have very exciting phase 1/2 studies. And then when you get to a phase 3 study, combining with standard chemotherapy, the drug doesn't do anything more beyond chemo plus placebo. So we essentially have five active chemotherapeutics, of which the response rates, even in the frontline untreated setting, are really only as high in like the low 30% range. And then in the second line setting for folks who are somehow still fit enough to get treatment beyond frontline chemotherapy, those response rates are usually 15% or less. So anytime you have a refractory patient population with pancreatic ductal adenocarcinoma that's achieving response rates, as in this case, in the 30 to even low 40% range, it's quite striking. And we think that's because we found an actionable target, which again is only expressed in this very slim minority of patients. But if you find one, the responses can be quite high and quite durable with a regimen that, compared to our multi-agent chemotherapy cocktails is fairly easy to tolerate with the exception of some infusion reactions and some GI toxicity.
Yeah, the overall response rate in the NRG1 study of Zenocutuzumab was 30%, and this was really across different tumor types. And these are rare events. We're still in the nascency of understanding these fusions. There are a lot of different fusion partners, a lot of different breakpoints, and that heterogeneity probably matters in some way. But at this early stage in effectively targeting NRG1 gene fusions, a response rate of 30% is, I think, fairly meaningful. In pancreatic cancer, actually, the response rate was a bit higher at 42%. Balance that with a very favorable tox profile. And as you mentioned, Ben, very durable responses. The median duration of response, 11.1 months. So just shy of a year, durable, meaningful responses for a notable subset of our patients really make this an impactful drug.
And usually, it's the opposite. Usually, pancreas cancer is the lagging indicator in terms of response. In other trials, it's true in microcellular instability, high pancreas cancer, where immunotherapy works, but for whatever reason, it works the least well in pancreas cancer. And taking a step back, mechanistically, so this is a drug that's a bispecific between HER2 and HER3, so kind of a unique mechanism because we think these NRG1 fusions, the fusion product is typically a transmembrane protein that's anchored, and then the NRG1 is now aberrantly kind of attached to it in a way where it can constitutively activate the HER3 pathway. And so what this molecule does is it binds on one hand HER2, but on the other hand HER3, and kind of disrupts this NRG1 HER3 mediated signaling that leads to downstream constitutive activation of specific oncogenic pathways. So it's an interesting and somewhat unique mechanism of action that really, requires this fusion to be there. There are other drugs that have been developed to target HER3 directly, but this, mechanism seems to be the most beneficial based on, clinical data we've seen so far.